region, and I use two specific probing regions. And a probing region will probe a specific structural characteristic of the RNA. And the first characteristic that I'm looking at with the shape region for selective 2' hydroxyl acylation analyzed by primer extension is the level of flexibility of a nucleotide. You have here a shape region, and if this nucleotide if a nucleotide is flexible, it will react with the 2' OH of the ribose. So it will react with the ribose moiety, meaning that it will react with all the 4 nucleotides, A, G, C, and U. And when a nucleotide is flexible, it is most likely that it will be also single-stranded. So it will create a 2' O adduct upon reaction, and this groupment, the addition of this groupment will cause the RT to stop further, and it is how we will be able to map these sites. And why the shape hasn't been used for, prior to recently for the probing of RNA structure in vivo is because it has a too short half-life, but from few seconds to few minutes. But a new generation of shape has been reported recently with longer half-life, and they are cell permeable, and it allows us to modify RNA structure in vivo. And the second compound is the DMS. So the DMS will look at the accessibility of specific groupments, and there are two that I want you to remind, which are the nitrogen 1 of the adenine and the nitrogen 3 of the CPD. And if these are available, the DMS, the methyl sulfate, will add a methyl group that will also cause the stop of the RT during a reverse transcription. And these groupments are on the Watson-Crick edge of the nucleotide, so when they will form Watson-Crick base pair, they will be protected by hydrogen bonds and won't be modified. And the DMS is also cell permeable and allow the modification of RNA in vivo. So I have my two probing regions. So now how do I treat the embryo to probe the RNA either in vitro or in vivo? So first in vitro, I take an embryo at a precise state that I want, I extract the total RNA, and it is this total RNA that I pre-fold into a pseudo-physiological buffer that contain DMS or NAI, which are our probing agent. And then I stop the probing reaction by quenching and then analyze what are the modifications either by primer extension or library preparation. And for the in vivo, it is pretty similar, except that the embryo at a precise stage will be backed in a solution that contains our probing region, and then it will be quenched and the RNA will be extracted, and then the modification will be analyzed by primer extension or library preparation. So first, to optimize all the conditions, so the concentration of probing region, the time, the concentration and efficiency of the quenching agent, I had to test it using a standard RNA probing technique, which is primer extension, where we have our RNA that is modified, and then by using a specific RT primer, 5' label with P32, we can do an RT. The RT will stop at the modification site and produce a cDNA. But because we have more than one molecule in solution that will be modified at different places, we have an array of cDNA, and when we separate them on a page denaturing gel, we can see this kind of pattern where we are able to map where the RT stopped with the sequencing line here and quantify the level of modification by looking at the intensity of the bands, and with that we are able to normalize and do some bar plot analysis, or to look how our data fits a secondary predicted secondary structure model, or even to use our data to generate more reliable predicted secondary structure. So I use this using the 5S ribosomol RNA because it's an abundant RNA in my case to optimize the probing condition. And the first one was for the shape. So when I treat with shape in vitro, I was really curious to see if my compound will work because they are not available, commercially available, so I had to synthesize them myself. So thanks to the Jorgensen lab in the chemistry department who helped me to achieve that. And I was glad to see that indeed my shape compound seems to be efficient because it modified the RNA when they are added, but when I add the bertamecaptoethanol, which is my quencher, prior to the addition of the shape region, I can see no more modification. And when I look at where those modifications occurs, I can see that they are indeed generally in the predicted single-stranded region that should be more flexible and prone to react with shape region. Now, for DMS in vitro, again, I found a good concentration that allow us to have a good probing in vitro, and again, the results that we obtained for the in vitro probing correlates with the secondary structure that that is predicted for a 5S-ribosomal RNA. And more interestingly, when we look at the in vivo probing of the DMS, we can see that a lot of bands disappear. Actually, almost all of them, which makes sense because, as you know, a 5S-ribosomal RNA in vivo is in a complex called a ribosome and interact with a lot of ribosomal RNA and ribosomal protein. So it's normal that most of their its groupment becomes less accessible to probing to DMS. So this suggests really that our condition for in vivo probing looks to be able to probe and keep the integrity of RNA protein complex and RNA complex. And actually the two bands here, the only two bands that we see are the ones that are at the extremity of the stem loop. So we have a good condition to probe RNA in vitro and in vivo for DMS. So how we can now map all those modification sites on a transcriptome-wide manner. So during my first year, I developed a technique that is really similar to what the Bibilaka and Asman lab have reported in past October in Nature, and they call it structure-seq. So I will use the same nomenclature. So structure-seq is here you have a modified RNA with DMS and a negative control where we have no probing region. And instead of using a specific primer for the RT, we use a degenerated primer with 9 positions that are degenerated, and our RT oligo also contain a known sequence in its 5'. And this known sequence would be important to generate the library, the PCR, after single-stranded DNA ligation. So we extend, and the RT will stop at modification site, and when we prepare the library and send for deep sequencing, we're able to map those modification sites. And here you have, for example, for the C. And we do the same thing for the negative control because there are 3 reasons why the RT can drop us. First, it reaches a modification site, or it reaches the end of the transcript, which makes sense, or it reaches RT drop-off prone region that can be a structure, highly stable structure, or a repetitive element. And they are actually quite frequent, so it is why it's really important to have a negative control in this case. And this negative control will be used for subtraction after normalization for the depth of sequencing, and then subtracted to get the normalized reactivity for the DMS condition. And because we have more than one molecule, again, we will be able to have a profile, obtain at the end a profile of reactivity for each nucleotide inside the transcript. And this is also true for shape compound, but just to remind that shape can modify any of the 4 nucleotides. So here, for example, it's a T that we will also, from which we will normalize and also subtract from the negative control RT drop-off, and then we will also obtain a profile, but at each nucleotide. So the library that I built first were coming from a 2 hours post-fertilization embryos, a stage also known as 64-cell stage, and this stage is right prior to zygotic activation. So we will have a snapshot of the RNA structure throughout the transcriptome right before one of the more critical events of early embryogenesis, the activation of the zygotic genome. And I built a library for DMS in vitro and DMS in vivo, and also shape in vitro, and we have more depth of sequencing for the DMS libraries, and with the depth of sequencing that we have, we have over 10,000 transcripts for which we have more than 50% coverage on A and C for DMS, and for all the nucleotides, more than 2,000 for shape. So we have a fairly good number of transcripts in which we have enough information to study them further. And one of the first things I was curious to look at was the nucleotide distribution of the reads that we have, that we mapped, because they should be enriched in A and C in the DMS treatment, right? So here you have the distribution throughout the transcriptome for each of the 4 bases, and when we look at the DMS data, we can see that for DMS treated samples, actually more than 86% of the reads that we map correspond to a C or a corresponding to the modified base. While for the in vitro one, the shape one, sorry, we don't see this distribution. We see a more even distribution because this compound can modify the 4 nucleotides. And just to mention that I show here only DMS in vivo, but the in vitro is the exact same thing. So I showed you some primary extension on the 5S ribosomal RNA. And here, these are normalized values. So we can theoretically compare the intensity of the peak between 2 lanes. And we can see that the in vitro, again, is really more accessible than the in vivo, which makes sense because it's in the ribosome. And actually, when we look at the highest peak in vivo, it corresponds to the same highest peak that we have seen with using primary extension. So the data at this level seems to correlate. And if we look for another ribosomal RNA, here the 5.8S ribosomal RNA that is known to interact with in plants the 25S, but in mammals the 28S, and this is an interaction that is conserved throughout organisms. And here the same group that developed structure-seq probe this 5.8S in vivo, and it is the data that they obtained in red, the nucleotide in red being more accessible in DMS in vivo. And when we compare with our data, well, again, we are able to see a big discrepancy between the DMS in vitro and in vivo, while the in vitro probing of shape seems to correlate pretty well with the DMS in vitro link. So again, suggesting that we keep this interaction when we probe the RNA in or in vivo condition. And when we look at the nucleotide that become more accessible, and that they are accessible, in DMS in vivo, well, they correspond to the nucleotide that are in the predicted single-stranded region in this conserved interaction. So these were endogenous control, but I told you that we can inject almost whatever we want in an embryo. So I injected exogenous control, and when came the time to decide what type of RNA structure to inject as a control, I thought about a side project that I had that was using a tRNA cassette. So tRNA might be probably one of the better known RNA structure with this really well-known cloverleaf structure. So I have this cassette, and in this cassette it will fold into the cloverleaf structure, but in the anticodon stem loop we can actually insert an RNA of interest, and it will help to allow a proper folding of this RNA when you want to express it in vivo. And in this case it's spinach, that is also known as the RNA GFP, but I don't want to talk about it too much because it doesn't fall into the program of this talk. So just to let you know that I inserted this tRNA spinach cassette in the 3' UTR of a transcript, so it will be a good control also to look at the ability of structure-seq to get structural information inside a longer mRNA, a longer transcript compared to the ribosomal RNA. And one of the first things that we can see that differ from the profile that we have seen for ribosomal RNA is that in this case, the DMS in vitro and in vivo are in really good agreement one with each other. And again, the shape also is in agreement with the DMS compound, which makes sense because we don't expect any protein or molecule or RNA to interact with our structure here, tRNA spinach cassette. And when we look at on the predicted secondary structure where those modifications are, we can see that again they fall into the region that should be single-stranded. So structure-seq seems really to give informative RNA structure information, and or in vivo treatment seems to be really reliable to probe the complex, the RNA in its native form, for example, keep the interaction that they have with protein and other RNA. So this I will skip.